All right, so we're going to go ahead and talk about plasma membranes and their structure. And they're really the outer layer of a cell, and it kind of keeps everything in to maintaining their internal environment, and it keeps other things out. And so we're going to spend a lot of time talking about how they do that. So, the first part is we're going to look at the fluid mosaic model. We're going to look at its structure. And so the reason why it's called this is it's fluid because the phospholipids are in constant motion. And the proteins that are in the cell membrane actually can drift around. And then the reason why it's called a mosaic, I'm going to kind of write it in there, is there's all kinds of different things. And if you look at this picture, even you can see that there's all kinds of different things. So it's a mosaic of different molecules or different structures. So mosaic of a variety of molecules. So let's go ahead and dive in and look at what some of those are. So let's actually start with the main structure, which is, as we all kind of know, is I'm going to circle it in purple in this one right there. So that is our phospholipid, and they make a bilayer because there's two of them. There's one on top and one on the bottom, and they can flip places. They can flip side by side. So there's our phospholipid bilayer. And it's obviously made out of phospholipids. So they're kind of always depicted like that with the circle and the two lines. And so that circle part is what we call the head, and that is hydrophilic, or also known as polar, right? It has that has a phosphate group that makes it that polar, and it's attracted to water. And so it wants to stick outside into the aqueous cell environment or outside into the extracellular matrix, the stuff outside the cell. And then the lipid part is there's two fatty acid tails. So we have two fatty acid tails that make up the tail part. Right, tail, what I'm writing about. And they're the opposite, right? They're hydrophobic. And so they're also, another way to say that is they're nonpolar. And what's nice about that is that acts as a really big barrier, right? It makes it so other molecules can't get in. We'll talk about that a lot, what can get in and what can't, but that's the reason why. And we also know about saturated fats and unsaturated. So if they're more saturated, those tails, there's more kinks in them. And so there's more space between the fatty acids, so they're more fluid. It's one of that fluid parts of the fluid mosaic model. Okay, so that's the main structure, and that's what keeps it semi-permeable. We'll talk more about that in a different video. Another thing that we might notice on here is, let's do these guys right here. These little red things, these are terribly drawn in this picture, but we're going to use them we're going to note them anyways. And the reason why they're terribly drawn is because they're not showing their structure at all, but that is cholesterol, which is a steroid molecule. So it is also a fatty acid or a fat, hence it's hanging out by the fatty acid tails. And cholesterol helps maintain and restrict membrane flexibility. So it kind of helps the rigidity of the membrane. Okay, that's what our cholesterol does. And then let's notice this big, huge protein that's going all the way. It spans across this phospholipid bilayer. And then also we have this protein here. Both of those are the same types of proteins. They're integral proteins. So I'm going to write that guy, integral proteins. They are embedded in the membrane, as you can see. So part of their structure is hydrophobic, like the inside part, and then the outside part is hydrophilic. And they we're going to see them do all kinds of things, but a lot of times they're transport, they're carriers, They also act as receptors. We're going to see them lots and lots and lots. Okay. And then the other one, I'm going to do it in the light green just because it's also a protein, is this guy that's kind of sticking off. That is what we call a peripheral protein. And it's usually they're found on the inside of the cell, and they're points of attachment to the cytoskeleton. They kind of help anchor things. Remember that inside our cells, we have this really complex cytoskeleton or network of things to move stuff around. Cytoskeleton. And then we are down to just these little sticky outies, as I like to call them. So what color haven't we done? We haven't done a blue. So I'm going to kind of circle these guys. These guys right there. Both of them are oligosaccharides. So if we're hearing the word saccharide, we can think of what? A sugar, you should be saying to yourself. So they're surface carbohydrates, surface carbs. I'm getting lazy. And so that you can see they have some short branching chains, and they're a lot of times involved in cell-to-cell recognition. So they're how a cell can identify itself to another cell, and say, hey, I'm from you. We're the same person. We're all good. It could also go the other way and not be so, and be like, hey, you're a foreigner, get rid of you. And you can begin to attack it. And so there's actually two kinds, and we can see them here. This first one, I'll put a 1 next to it, and then I'm going to write a 1 right here. So number 1 is a glyco, that's the sugar part, lipid. And you can see it's attached to a phospholipid. Okay, so it's attached to a lipid. And then I'm going to put a number 2 right here because you guys can probably see that it's attached to a protein. So we're going to call it a glycoprotein. And it's going to be attached to a protein. Cool. So those are all the parts of the cell membrane, and we're going to see how they function in other videos.